On to the last chapter, last section of the chapter. We'll be moving up in time. So this work was done around the turn of the century in 1900s. Papers were published about 1910, 1915, and the work was done by Thomas Hunt Morgan. And in his lab, he worked primarily with fruit flies, Drosophila melanogaster, the same fruit flies we're doing our experiments in the lab. And there's a lot of great reasons to choose this as a model organism. The flies are more sophisticated than plants, so they're a higher organism. They're animals. They reproduce very quickly. They are easy to keep tons of them in the lab, and they have some pretty strange characteristics that are easy to track when you introduce some mutagenesis. So basically what Morgan's group did was to take flies and treat larvae or eggs with mutagens, which would randomly mutate things in their genome. And we'll start with an explanation on these particular flies. So these are called wild-type flies. They have nice red eyes, fluffy wings, kind of a grayish body, striped pattern. And then the mutants that he's going to work with in this particular experiment that we'll look at first have crumpled up wings called vestigial, and that'll be abbreviated VG. And they have a really dark body, so that was called a black body. Both of these traits turned out to be recessive. So here we have a fly that's labeled lowercase b, lowercase b, lowercase vg, lowercase vg, meaning that he is black and vestigial. The wild type or normal one. The nomenclature that this textbook uses is a little bit different than actually what Morgan used. They simplified it a little bit here, and we can use this, but I just want you to be aware of the fact that you may see it in other places with a different name, and I may revert to that because that's what I've been using for a long time. So what you may see is that for normal flies, it has a lowercase b plus, with the plus meaning you're normal. Lowercase b stands for the black gene and that it's recessive, but the plus means you're normal at that loci for that allele. You could also be a normal body if you're a black plus b fly. So we mate these two flies, and the question could be posed, do alleles for different characteristics always assort independently? Now remember, Mendel did that. He made that his second law, the law of independent assortment. And that's true when genes are on different chromosomes. When Morgan moved to the fly, he moved to a different number of chromosomes in an organism. The flies have 4 chromosomes. They have 3 autosomes and 1 set of sex chromosomes. They have an X and a Y, and that's going to be the other thing that Morgan figured out that Mendel didn't really understand because he worked with pea plants. So, we're going to cross these two flies, and let's just look at the outcome, and from that we'll be able to explain what Morgan found. So he crossed that are heterozygotes. So he's heterozygote at the body color position, heterozygote at the wing position, with a completely recessive. This is called a test cross, actually. Whenever you cross something with a double recessive or a complete recessive, it's a test cross. And the outcomes are this. We could do the Punnett square. The only gene gametes that this guy can give are lowercase b and lowercase vg. And then there are 4 possible gametes from this one, right? We can have the wild type with the recessive and so on. So we get out flies that are heterozygous at both positions. We get out flies that are homozygous recessive at both positions. And these look like the parents, right? You can also get mixtures, right? You can get a heterozygote for body color, homozygous recessive for wings, homozygous recessive for body color, heterozygous for wings. Mendel would predict that these would happen in equal amounts. We'll draw that Punnett square up on the board in class. However, what happened in the experiment was something surprising. They had a disproportional numbers looking like the parents and a smaller number as recombinants, because basically we're mixing and matching those chromosomes. They're resorting. And we basically can say from this, no, those two genes are not sorting independently. They seem to be linked, but they're not linked all the time. So what's going on? Remember, when crossing over happens in the meiosis, we can look at, this is the total recessive and this is the heterozygote. Or no, I'm sorry, these are both homozygous. They're going to cross over part of the time. and then you end up with a mixture. So we have one chromosome that looks like the parent, one chromosome that's a mix, one chromosome that looks like the parent's, one that's a mix. The frequency that that happens depends on how far away they are from each other. End up with that meiosis too. So how does that look with the flies? It turns out that the farther apart you are, the more you cross over. The closer you are, the less you cross over. So Morgan's group figured this out, and they crossed a lot of different other mutants. They had mutants that had yellow bodies. They had ones with white eyes. Actually, that's a really bad example to put on here. Ignore that for right now. I'll just say the eye color. Here's another eye color mutation. Vermilion's kind of orangey looking. Mini wings, M, and then rudimentary wing, which isn't even a vestigial wing, it's just a bud. And the numbers that are going on here are basically placing those genes along the length of a chromosome. So at the turn of the 1900s there, 1910 or so, they didn't have sequences. They didn't even know how to sequence a gene. Now we have the whole human genome sequence, so this is like, yeah, we could map anything we want. We just look at a database. But at this time, it was pretty interesting that they were mapping that certain characteristics were at certain locations on the chromosome, and they were always there. And the way they got these numbers is by noting how frequently they crossed over. So the distance between this yellow is at 0 and vermilion at 31 means that if you compare crossing over between these two genes, if you do a dihybrid again, 31% of the time they're going to cross over. And that gives it a placement on the line. The same could be said, the distance between these is only 3, so they're not going to cross over very frequently. 3% is not a very high rate of crossing over. 34 to 58, what's that, 24, that crosses over fairly frequently, so 24% of the time. So I'm saying this percents. Where do they get those numbers from? They get that from the recombination frequency. So again, if you take a heterozygous wild-type fly and cross it with a double recessive, and again, we'll put that Punnett square up on the board, but here's the data right here. They have a disproportional number of parental phenotypes, so they look like the parents, and then they get this 391, if you add these two values together, of individuals that represent crossing over or recombinants. So they came up with a formula that if you divide 391 by 2300, which is the total number of offspring, you can say that they cross over 17% of the time. And that's exactly how they set up the map between B and VG. So if they were to lay these guys out on the chromosome, they would put a number of 17 in between these two letters and say that's 17 centimorgans, or approximately 17 units away from each other. So the fly guys did a lot of this, got a lot of satisfaction out of figuring out sort of like a map for the fly genome. Then the next really big thing that Morgan's group contributed was to understand that there are sex chromosomes. So Mendel had no clue about this. His pea plants do not have sex chromosomes. It's only when you get to animals and that you start seeing these. Different animals can have different numbers of sex chromosomes. They have them different ways. The flies happen to have an X and a Y similar to ours. This is a human X and Y chromosome. You're going to see an image in a minute about the flies. So X's have a lot of information on it. Here would be the sister chromatid of the S chromosome, X chromosome, and here's the Y. It's a lot smaller. Y basically has all the information on it that you need to be a male. So this here is a male. The picture in your textbook shows you the genome for the flies. A little funkier setup, but anyway, X and Y for males and females. So here's what Morgan figured out. They were doing their mutation and they found flies that had white eyes, and that's here. And you're going to see some of those white eyes. You've already seen them in your crosses, and we'll be able to follow this. I'm looking over there at the flies when we start counting them in a week or two. Again, you can do a couple different ways to do the nomenclature. Whenever I do Punnett squares for this and I write stuff up and I'm solving problems and thinking about it, I always am sure to put the Y in as a placeholder. So when I say that the genotype for a male with white eyes, I'm going to give it the lowercase w because this is recessive, and I'm going to put a placeholder in for the Y because he doesn't have two copies of that W. If a male has red eyes, again, using Morgan's convention, which this current textbook doesn't quite use perfectly, you would say that white eyes is recessive, but he has a plus there, so he's normal at that location, and then he has a Y, so he has red eyes. A female with red eyes could be a W plus W. or she could be a W plus W plus. The way that they note it here is not so bad either. So they're saying on the X chromosome there's a gene for red eyes. This one is dominant, XX. And then they're saying this male has the recessive, which they're putting in as a lowercase r, but we can call it W if you want. And it's X lowercase and then Y. So what happens if these guys make gametes? So the sperm is going to be one X chromosome in one sperm and then one Y chromosome in the other sperm. The eggs are going to get the same thing. They both are getting a red gene on the X chromosome. So if you make a red-eyed female with a white-eyed male, none of our flies will have white eyes because the females get 2 copies of the X chromosome and they're carriers. The males only can get an X from the female, so they are going to be normal red eyes. So we can do yet another cross. What if we cross a white-eyed female? They do exist. You just have to have 2 copies of the white gene. So here she is over here, and then she's crossed with the sperm from this fly who's normal, so he has red eyes and he has Y. So all the girls are going to be carriers, and then all the boys will have white eyes. So what if you cross these 2? Can you finally get a white-eyed female? So we'll do that Punnett square in class because you can get white-eyed females if you cross a carrier with an affected male. There's a lot of cool things you can do with that. There's a lot of X-linked diseases. So these are called sex-linked diseases or sex-linked disorders. Let's see if I could put a text box in here. Hemophilia is caused by a defect in one of the genes for clotting your blood. You bleed a lot, and the gene for clotting is called factor X. It exists on, factor 8. It exists on the X chromosome. So there are a lot of males who can get hemophilia. Also, red, green, color blindness. The 2 genes, one for red and one for green, they're actually separate ones, are on the X chromosome, so it's more common for males to get that. And then there's a muscular dystrophy that's more prevalent in males. Basically, if you're a guy, this one doesn't have it. Let's look at this one. You only get 1 chance of having good genes. You only get 1 X chromosome. So if you have a defect on it, you get the disease. You have nothing to complement it. Whereas females can be carriers. They could be heterozygotes and they could still be all right, and then they're not going to see the effects of a gene. So there's some more consequences of having the chromosomes, and one can be explained by my cat. So Kitty arrived in our house when my daughter was only 2 years old. She jumped in the backyard. That's why she has the name Kitty. We tried to find Kitty's owners. We walked around with signs and drove around with the wagon to show around for Kitty. Nobody claimed Kitty. So finally took her to the, told my coworkers about the story that now we have Kitty. Haven't really touched the cat, you know, she was kind of nervous. Oops, I gave it away. Cat was kind of nervous and couldn't really get close to her. But as I was relaying the story to the guy I worked with, he goes, oh, well, it's a girl. And he's kind of an arrogant guy, so I just figured he was just being a know-it-all. But he actually was right. All calico cats are girls. So it turns out that the genes for their coat color are carried on the X chromosome. So you can either have the black gene, you can have the orange gene. Oops, orange gene. White turns out to be a totally different story. But you can carry the black and the orange genes. So in order to express both black and orange, you have to have 2 X chromosomes. If you're a boy cat, you can only have black or you can only have orange. You can't have them both. And as it turns out, early in development, some of your X chromosomes are basically decommissioned. They condense into what's called a Barr body. Only females get this. So you really don't need 2 copies of the X. All the boys exist just fine with just 1 copy. So girls have to disable one of their X chromosomes. And that happens very early in development. So basically it sets up a mosaic pattern. And then all the progeny cells, if they're expressing the black gene, then all those cells are going to have black. If the orange gene on the X chromosome is the one that survives, then all those progeny cells will be orange.